أهلا بيكم في دروس الأحياء للصف الثالث ثانوي لغات. Today's lecture we will start discussion about DNA and and about the structure of DNA. So let's begin. Chromosomes have two substances in them, DNA and proteins, but which of them carries the genetic material? Since the proteins are made up of تشكيلة من 20 amino acids, different kinds of amino acids, monomers, while DNA معمول بس من 4 nucleotide monomers. يبقى الدي ان اي من 4 نيوكليوتايد مونومرز، لكن البروتينز بقيه البروتينز من تشكيله من 20 different amino acids. فكانت الناس فاكره ان الجينيتيك ماتيريال carried by proteins، لكن الدي ان اي هو contains the genetic information زي ما ثبت بعد كده. طبعا discovery of the DNA is the genetic material خلى نشوء علم اللي هو الموليكولر biology. اوكي، كأول evidence يعني على genes were made of proteins. هندرس حاجة اسمها bacterial transformation. Bacterial transformation, scientist named Griffith studied the bacterial pneumonia, and he could have 3 strains of or different strains of the bacteria pneumonia that could be transformed into genetically different strains. Like, for example, he had one of the 2 strains where virulent bacteria, he called it the S type. As you can see, when injected in mice, the mice were killed. The other strain was non-virulent bacteria. He coded it with the letter R. So when injecting the mice with it, it was non-lethal to the rats. Okay. At one experiment, he injected non-living virulent bacteria, S type I mean, but non-living virulent bacteria with living non-virulent bacteria, which is of the type R. What happened? He injected it into the mice, and the mice were killed. And by dissecting the mice, the kind of bacteria in them was a living virulent bacteria. He did not inject them with a living virulent. He injected them with a non-living virulent bacteria. Remember, together with a living non-virulent bacteria, but the result was living virulent bacteria. So there must be a kind of transformation, bacterial transformation that happened that led to the existence of these living virulent bacteria. A scientist called Avery and colleagues isolated the virulent bacteria from the virulent bacteria, a material that was responsible for the genetic transformation. This material was DNA. Okay, so the experiment concluded that bacteria absorbed DNA of another strain and acquired the characteristics of this bacterial strain. But how did it absorb it? It's not known. Some other scientists debated that the DNA used was pure enough and it was mixed with proteins, and why not the protein were not carrying, aren't the ones who carried the genetic information. But until they discovered the hydrolyzing enzyme called deoxyribonuclease, which can hydrolyze DNA completely but not RNA, and they use it to apply it on the transforming material, transformation did not occur. Why? Because the DNA was destroyed. So this proves that DNA carried the genetic information, or DNA is the genetic material. For another evidence that DNA is genetic material, the study of what's so-called the bacteriophage or phages. These are kind of viruses with DNA inside a protein coat, and the phage that will perform this experiment on or with consists of a DNA molecule and a protein coat, and the protein coat extends as in an extension as a tail. The tail, the bacteriophage uses it to attach to the bacterial cell wall. See that? To the bacterial cell wall. He allowed for the invasion of bacteria with the bacteriophage and measured what happens in the bacteria, inside the bacteria, in different time intervals, and after 32 minutes the bacteria burst out and 100 new complete phages emerged from the bacteria. This means that the bacteriophage, which is a virus, transmitted its genetic material inside the bacteria, and it must contain the virus genes. Okay, so to make to get another evidence, how about marking the constituents of the different components of the DNA and proteins? That is, DNA contains phosphorus, but the other proteins do not contain phosphorus in them. So that's one thing. Other proteins contain sulfur in them, but DNA does not contain sulfur. So if we marked the phosphorus and if we mark the sulfur and observe this experiment again, we will know how did the genetic material was transferred, through what? Through proteins or through DNA. And it will show, of course, that it is transmitted through DNA by measuring the radioactive sulfur and in the phage protein and the radioactive phosphorus in the phage DNA. Okay, this is how to prove that the DNA was the genetic material. The DNA actually entered the bacterial cell and pushed it to the side and build new viruses. It used the bacteria as a factory for new viruses. This also proves that the genes are composed of DNA. The genes are composed of DNA. But the question is, are all genes composed of DNA? The answer is no, because we have some exception to the rule, which are RNA viruses. They contain RNA, and genetic material in these viruses is the RNA. But this is very few strain. It's an exception to the rule that the most living organisms, DNA is the genetic material in nearly in all forms of life. Okay, so how about the quantity of DNA in the cells? They measured the quantity of DNA in different kinds of somatic cells and reproduction cells, and they compared them, the DNA in somatic cells and reproductive cells. Also, they measured the amount of proteins. The amount of DNA was equal among cells, but amount of proteins was unequal. So when comparing the somatic cells and reproductive cells, remember, from reproduction, it's the gametes, the haploid number. They carry the haploid number of chromosomes. So they carry the haploid number, or half the genetic material, half of the amount of DNA in the somatic cell. This is when you compare them. And this we know why, because there is a female gamete and a male gamete, and both complement each other to form the full diploid number of chromosomes organism. Okay, also the proteins are continuously being made and destroyed. not like DNA, which is very stable among the cells. To study the structure of DNA, any model of DNA structure must take into consideration the following: that the basic structure is, the basic unit is a nucleotide, and the nucleotide is made up of 3 parts: a 5-ring 5-carbon sugar, C, and a purine or pyrimidine base, and a phosphate bond. Okay, phosphate group connected with the covalent bond to the sugar's 5th carbon. This is the 5th carbon of the sugar. So again, it's a sugar, 5 carbon. To the 5th carbon is phosphate group and base, pyrimidine or purine base. The nitrogen base may be a single ring like the pyrimidines, which are the thymine and cytosine, or a double ring like the purines, like the adenine and guanine. This is a double ring, so this is either adenine or guanine. Okay. When the nucleotides are linked together in a strand of DNA, the phosphate molecule attaches to the 5 carbon of sugar of one side and to the 3 of the adjacent side to form the sugar phosphate backbone structure. This is the sugar phosphate backbone structure. See the phosphate groups to the 5 of one side and 3 of the other side, 5 and 3 of the other side, adjacent side, and so on, and form, 2 sugar phosphate backbones with opposite direction of the free hydroxyl group. See, this is the free hydroxyl group at position 3 at this side, and there's here the phosphate group at the 5. But the opposite side is totally the opposite. This is the hydroxyl group, and this is the phosphate. See that? In each DNA molecule, the number of nucleotides containing adenine must equal the number of containing thymine, because adenine is coupled with thymine. And the number of containing guanine equals the number containing cytosine, because cytosine is coupled with guanine. See, a double bond between the adenine and thymine, and a triple bond between the cytosine and guanine. Okay. Evidence about the structure of DNA is simply take a picture of it. How to take a picture of it? By recording the pattern of scattering of X-ray off the molecule of the DNA. It can give you a great idea about the purity of the or the highly purified DNA and how it looks. A scientist called Franklin tried this to see how the DNA looks like. She confirmed that it's twisted and the bases are perpendicular to the length of the fiber, and the sugar phosphate backbone is on the outside and the bases is on the inside. See that? الشوجر باكبون موجودة على بره، لكن البيسز موجودين ناحية جوه. يكونوا مع بعض هيلكس زي حلزون يعني، ولازم بقياس الدايمتر بتاع الهيلكس ده تلاقي بيتكون انه more than one strand of DNA. Actually it's 2 strands of DNA. 2 British scientists بعد كده جم بعد فرانكلين and they confirmed that it's 2 strands of DNA. عبارة عن زي اللادر يعني، زي ما انتم شايفين. دي اسمها الرانجز بتاعت اللادر، يعني الدرجات بتاعت السلم، الرانجز. اوكي، شايفين كل رانج متكونه من coupling of 2 bases either cytosine مع جوانين او adenine مع thymine. adenine paired to a thymine وجوانين paired to cytosine. والpair of bases in each rung is held together by hydrogen bonds. دي اللي بينهم دي hydrogen bonds. 2 hydrogen bonds between الادينين والثايمين زي ما انتم شايفين هنا، و 3 hydrogen bonds between الجوانين والسيتوزين. ده اللي استخلصته او اللي استخلصوا الاثنين علماء اللي هم واتسون and Crick after studying the shape or the structure of the 2 strands of DNA molecule. Okay. Each rung is composed of one base of one ring and base of double ring. This means that the width is the same لأن نفس الحكاية. يعني في الرنج الواحدة في بيورين وبايريميدين، بيورينايا وبايريميدايا، و الرينجز عددهم واحد، يبقى كده في كل رانج يعني. فيبقى نفس الديستانس between one another and along the length of the DNA molecule. Okay, the 2 strands of DNA run into the opposite direction, as I told you before. At one side, here the phosphate is at the 5 carbon, and the hydroxyl group on the opposite side, and the same applies to this side. And the whole ladder of DNA is twisted with 10 nucleotide pairs per turn. تمام، عشان يكون سبايرال. عشان كده سموا ده الدبل هيليكس. الدبل هيليكس لانه مكون من 2 strands of DNA connected in the middle زي ما انتم شايفين هنا كده. Okay, I hope you got the maximum benefit from this lecture, and thank you very much for watching.